Claire, are we good with the screen?
Yes, I can see it.
Okay, perfect. Okay, well, hello everyone. Thank you for joining us today. My name's Lindsay Stone. I am the Plant Stewardship Coordinator with Amargosa Conservancy, and we are hosting a botany webinar series this summer. And so for our first talk in the series, we're going to be hearing from Allison Autry, and we have another webinar coming up in August, so stay tuned for those details coming up soon. And yeah, we'll have time for questions at the end of this presentation, but I think Allison's going to introduce herself, so take it away, Allison.
Yeah, thank you so much, Lindsay. And thank you all for being here. The Amargosa Conservancy is a really great organization, and I've loved my time getting to, spend time out in the Amargosa and research plants there. So it's really great to be able to talk to you all today, with this Conservancy webinar series. Today, I'm going to be talking to you all about orchids in the Amargosa with a particular focus on my study species, the Ash Meadows ladies tresses. Um, just a little bit about me first. I am a PhD candidate at the California Botanic Garden. I'm a researcher under Dr. Naomi Fraga's lab, and she is a really prolific researcher in the Amargosa, as I'm sure many of you know, super involved with the Conservancy. So she kind of introduced me to this region. I've been a lifelong orchid enthusiast since, before I joined into the grad program at Cal BG. I've worked with both native US orchids out east. I've been an orchid enthusiast growing, the hobbyist tropicals that a lot of people enjoy. But these days, I'm really a native orchid conservation focused researcher. And currently, for my dissertation research, I'm working on rare orchid research based in California and Nevada, including the Amargosa region. First, I wanted to talk to you all today about what it means to be an orchid. I know it might take a little bit of convincing for some of you to believe that these plants we have out in our deserts in California and Nevada are the same type of plant as what you might find in tropical orchid collections at a botanic garden. So we're going to kind of discuss what these plants actually do have in common. Then I'll tell you all what native orchids can be found in the Amargosa region before introducing you to the plant of the hour, which is the Ash Meadows ladies tresses. After we discuss Ash Meadows ladies tresses a little bit, we'll get into the threats facing this species as well as other native orchids in the United States. And then finally, I'll share with you all the research that I'm currently doing on the Ash Meadows ladies tresses and how that research can help with the conservation of these species. So first, what does it mean to be an orchid? There's a lot of floral variety in this group of plants, maybe some confusion among the general public on what makes an orchid an orchid. The showy flowers are what they're famous for, but that's not necessarily something that all of them are going to share. The dictionary definition means that an orchid is any member of the family Orchidaceae, which is, a little bit of a tautology to define it that way. It involves perennial, epiphytic, or terrestrial plants that have showy three-petaled flowers with the middle petal enlarged into a lip and differing from the others in shape and color. So, what does that actually mean? Simplified, orchids are all members of the same family of plants, and they're going to share certain identifying traits as a result of being part of this family. Many plants that people associate with orchids are showy, brightly colored, and tropical, but not all orchids fit this description, and especially not the orchids that we're going to be finding in the US. Taxonomically speaking, a plant family is a group that's larger than genus, smaller than order, and plant family members are going to share genetic similarities and vegetative traits in common that plants outside the family do not. These shared unique traits are called synapomorphies. So when we're looking at the orchid family, what morphological traits are the synapomorphies? Mostly, you're going to find those in the flower. So when you look at an orchid flower, one of the most distinctive traits that really makes this family a unit unto its own is the fused column. So the anthers, where the pollen would be found on a normal flower, and the stigma, where the flower would receive pollen in a typical flower, are all going to be fused into one column in orchids. They have another unique trait in their floral morphology, which is the fact that their pollen, instead of kind of being loose and grains that can easily be dispersed on insects' backs or on the wind, are going to be kind of packaged into these sticky units called pollinia. Orchid flowers also have specialized labellums, which is where one of their petals is modified into this really showy lip. They're still going to follow the generalized monocot floral formula, being monocots in that group of plants with, grasses and the like. So they have three sepals and three petals, one of which is modified into that showy labellum we were discussing. So all orchid flowers will follow roughly this formula, even if there's some variety in, the degree of their showiness or how large the flower may be or the coloration and that type of thing. Reproductively speaking, there's one more big synapomorphy in the orchid family that's actually really relevant to how they live their lives, which is that they have these tiny, tiny fungi-dependent seeds. So these seeds lack endosperms, which is the nutritive tissue that's commonly found in seeds in this area of plants that are responsible for providing the embryo within the seed with enough energy for germination. The benefit of foregoing this endosperm for orchids is that they have these really small seeds that are wind dispersed. It also allows them to use fewer resources to make more seeds, so they get kind of more dice rolls at having an individual that survives to the next generation. However, the downside of this is obviously the lack of nutrient means that the embryo cannot really germinate in a self-sustaining way, and they instead require mycorrhizal relationships or relationships with symbiotic fungi to provide the seeds with the nutrients that they need to germinate successfully. A little bit more about the orchid family before we get into our local orchids here in the Amargosa. There are actually over 29,000 species of orchids worldwide. So this is a really diverse group of plants. They're found on all continents except Antarctica. And if you're thinking of orchids as a tropical plant, you're right in a lot of ways. Most of the orchid diversity in this family is found in northern and South America, Central America, and tropical regions of Asia and the Pacific Islands. However, we do still have orchids here in the United States, even if we don't have as much orchid diversity as some other regions of the world do. We have over 200 species of orchids native to the United States. So, what are the orchids that we can actually find here in the Amargosa? The first orchid that you can find here is probably one of the most common orchids in the entire Western US, and certainly really the only orchid you're likely to encounter with any regularity in the Mojave Desert or the Great Basin Desert. It's Epipactis gigantea, the giant stream orchid. So this orchid, like I said, is really widely distributed and can even be found in our desert regions. It does still require really perennial access to water. You're not going to be finding it in super dry areas. You'll generally find it in riparian areas, along streams or in seeps, or near springs in areas like Ash Meadows where we have these constant upwelling groundwater sources. This plant is pollinated by syrphid flies, but is a bit of a generalist. You can see a couple different kinds of pollinators on it. The flowers imitate a nest of aphids with their scent and appearance, and it's called the chatterbox orchid because that specialized labellum at the bottom of the flower on this plant is actually hinged, and so you can see it kind of wiggling or blowing in the breeze, and this might help it attract pollinators as well. And like I said, this is a plant that you can find in a lot of riparian areas in the desert, but it's especially widespread across Ash Meadows. The next orchid that we're going to discuss is kind of going to be the orchid that we stick with for the rest of today's webinar, which is Spiranthes infernalis, the Ash Meadows lady's tresses. So seeing a lady's tresses orchid in alkali seeps in the desert is a real novelty, and to understand why it's so shocking and exciting to find a plant like this here, I wanted to take a brief moment to introduce you all to the rest of the lady's tresses orchid. So that's the genus Spiranthes, and it's actually a quite widespread genus of orchid in the United States. It's widespread in the eastern United States in meadowlands and woodlands, and it also occurs in montane meadows and coastal areas in the western United States with pretty decent abundance in a lot of places. You'll find it all over meadows in the High Sierra. A couple of different species grow there. But Spiranthes are generally not extremophiles. You generally won't find them in incredibly dry or hot environments. They tend to like growing in these kind of mesic areas where they have decent access to water year round. You won't find them in very barren habitat either. They're generally in like gentle meadows, woodlands, places that look like, a habitable place to be a small herbaceous forb. But Spiranthes infernalis is unique. It's out here in the desert. It's in these alkali flats and in soils with really interesting composition. And it's just such a different environment to where you find most of the other lady's tresses orchid that it kind of makes it noteworthy. This is a newer discovery to Western science as well. Spiranthes wasn't even known from Ash Meadows until the late 1960s, early 1970s, and Spiranthes infernalis, the species, wasn't described to Western science until 1989 by Charles Chiviac. Initially, this plant was only known from Ash Meadows. That was the type locality where it was seen and described, and it wasn't known anywhere else. However, now it's known from two, potentially three locations in the world. It could be Nevada endemic. It might also have a range extension in Eastern California, depending on the results of some research that we'll get into later. And it's completely reliant on these groundwater-fed alkali seeps and marshes to eke out an existence in a place where otherwise precipitation would be far too meager to support its existence in that location. In Ash Meadows, Spiranthes infernalis can really be found across the refuge in areas with suitable moisture levels, and also just outside of the refuge itself. There are at least 500 total plants around the refuge and at least six genetically connected subpopulations. As many of you know, Ash Meadows is a challenging place to be a plant. The climate is quite extreme with summer highs up to 120 degrees Fahrenheit and winter lows below freezing. Evapotranspiration in this area greatly outpaces the average rainfall that the region receives. And so the Spiranthes and Ash Meadows are really reliant on being nestled in spots where there's good groundwater expression to be able to survive. As a result, these plants are really dependent that this environment remains stable and are vulnerable to outside threats, including anything that could result in groundwater depletion in the region, such as upstream mining projects or the proposed Western solar plan. The other location where Spiranthes infernalis is known in Nevada is out in Railroad Valley, which is a giant alkali wetland in the Great Basin Desert that's actually about 150 air miles northeast of Ash Meadows. Up until 2020, people thought that Ash Meadows was the only place in the world that you could find Spiranthes infernalis. But then Naomi Fraga was out on a trip in Railroad Valley and discovered Spiranthes there. Um, there are hundreds of miles of disjunct from Ash Meadows plants, so this was super unexpected. But there are actually, it turns out, a ton of Spiranthes thriving in Railroad Valley as we speak. There are three main subpopulations, with the largest two around Locks and Chimney Hot Springs in the valley. And in 2024 and 2026 surveys of the area, we found 400 to 450 blooming plants at Chimney Hot Springs and over 1,000 plants on Nevada Department of Wildlife and Bureau of Land Management land around the Locks Spring network. These plants are unfortunately threatened as well. There are a lot of proposed extractive industry projects in the area that could lead to groundwater depletion, and they are once again dependent on groundwater here, just like they are in Ash Meadows. And there's also a lot of cattle activity in the area, putting these plants at risk of trampling and herbivory. The final location where Spiranthes infernalis might be known from is Fish Slough, California, which is another alkali wetland just north of Bishop in eastern California. There's a very small population of alkali seep Spiranthes here, and they've proven to be a bit of an identification challenge. Spiranthes porifolia is a very similar looking and closely related species of Spiranthes known from the Eastern Sierra region in California. And initially, experts who examined the herbarium vouchers that were collected in Fish Slough thought that this plant was likely a member of the species Spiranthes porifolia. However, the habitat doesn't really fit. Spiranthes porifolia is generally known from much more hospitable areas. They're found at like mid to high elevation montane meadows in the Sierra, not really in these alkali lowlands where it gets this hot. And so the habitat really raises questions because it's a lot more similar to where you would find Spiranthes infernalis. But again, this is a pretty wide disjunct. It's not especially close to either Ash Meadows or Railroad Valley, and there are no populations of alkali seep Spiranthes between these locations that could serve as a means of connectivity. Another mystery kind of surrounding Spiranthes infernalis is what the pollinator relationships that this species relies on to reproduce are. Other Western United States Spiranthes species are somewhat generalist as far as orchids go when it comes to the pollinators that they'll affiliate with. In the Sierra Nevada, multiple bombus species have been observed pollinating these plants, but there haven't really been any good pollinator observations that I've been able to make with Spiranthes infernalis. I've been researching this species for about 3 seasons in the field now, and while I did one time get this really lovely picture of a butterfly hanging out on the orchid, I've actually never seen the pollinaria, like the pollen-containing sacs that are characteristic of orchids, getting moved from plant to plant. I've never even seen them dislodged from an individual flower. Still, in healthier plants, towards the end of the growing season, I see a lot of seed development and capsule development on these individuals. In many cases, like in the pictures I've included, every single flower up and down the inflorescence will eventually start to develop a seed capsule. So this raises a question. How are these plants developing so much seed if we're never seeing them get pollinated? There are a couple theories on how this could happen. Basically, the plant is self-pollinating somehow. Whether that's the pollinia coming into contact with the stigmatic patch and actually fertilizing the flowers and then developing seeds, or whether it's performing apomixis is yet to be known. Apomixis is actually an asexual reproduction method that plants can undertake, where seed develops without fertilization, creating genetically identical offspring. This does sound a little bit far-fetched and crazy, but it's actually a known reproductive strategy in other Spiranthes species, so that remains a pretty viable possibility for why Spiranthes infernalis can develop seeds so effectively despite not having any clear extant pollinator relationships. I also wanted to take a little bit of time to talk about why Spiranthes infernalis is so rare and threatened. So, this species is kind of threatened for two main reasons, the way I see it. One is because of its where, and one is because of its what. So, where Spiranthes infernalis is in Ash Meadows is an uncommon, threatened groundwater dependent ecosystem type. And this species, like many others found in the region, is completely endemic to this rare habitat type. Additionally, Spiranthes infernalis is an orchid, as previously discussed, and orchids are disproportionately rare and threatened compared to other plant species that they grow alongside throughout their range, not just out here in the desert. And so I'm going to take a little bit of time to talk to you all about both of these factors, the where and the what, and why they make Spiranthes infernalis so rare. So here's a map showing the richness of imperiled species in the United States. And you can see there are a lot of regions where we have high concentrations of rare and threatened species. So, Appalachia has a lot of really interesting plants. You can see California, floristic province, the plant biodiversity hotspot that it is really densely colored on this map. And then you can see this little tiny deep green dot at the end of the arrow I've put in here in southern Nevada, and that is Ash Meadows. So Ash Meadows stands out even on this, countrywide scale map of imperiled species richness because it's this really unique ecosystem that's full of imperiled species, not just my study orchid. So, Ash Meadows is home to species that are both intrinsically rare and incredibly threatened. So, when I say intrinsically rare, I mean these species would have never been especially common or widespread anyways. Things like the Amargosa niterwort and the Zeltnera spring beauties and the pupfish that live in the springs in Ash Meadows would have never been able to be distributed widely across the landscape. And they kind of evolved in these really uncommon ecosystems. And so they always would have been limited to these very small areas that have the appropriate habitat type for them. They never would have had especially high abundance outside of these areas or extended ranges. And so they're intrinsically rare, but they're also, unfortunately, now very threatened in addition to this intrinsic rarity. So, Spiranthes infernalis is facing the same threats as alkali wetlands throughout the Amargosa region, and they're facing the same threats that their non-orchid neighbors are. These threats include groundwater extraction and depletion, as well as hydrological alteration, solar development and geothermal energy development that are happening all over on public lands, lithium mining projects that are getting greenlit by the Bureau of Land Management, grazing and trampling by cattle that are in these areas, off-road vehicle incursion, pathogen introduction. Anyone familiar with the Amargosa has likely seen the die-off of the screwbean mesquites, which are a really important part of these habitats. Efforts to privatize public lands everywhere threaten Ash Meadows and the surrounding Bureau of Land Management lands as well. And then climate change, which can make an already really challenging habitat and climate to survive in as a plant even more inhospitable. Even though Ash Meadows National Wildlife Refuge is a protected area, it's still not completely safe. There are a lot of actively expanding mining operations in the Ash Meadows area, and the Conservancy is concerned about many projects that are being approved, permits that are being requested, exploratory drilling, proposed mining projects, and the like, all of which, while not within the boundaries of the refuge, are still within this watershed and can really alter the hydrological regime that Ash Meadows depends upon to exist and that all of its endemic species depend upon to survive. Additionally, Spiranthes infernalis is going to be at greater risk than even some of its neighbors because it's an orchid. Orchids are an especially threatened plant family everywhere that they're known to occur. And in the United States, more than 50% of our native orchid species are listed as threatened or endangered at some spot within their range, whether that's at the state or provincial level or at the federal level. These plants are threatened. Orchids rely on symbiotic relationships with both pollinators and mycorrhizae to complete their life cycle, and they tend to be more specialized than many other plant species and groups. Orchids kind of act as this canary in a coal mine. They rely on a lot of things to be going right within an ecosystem or a habitat to thrive. And so they can often be the first sign that things are kind of falling out of balance when they start to disappear in an ecosystem. And they're also, for this reason, one of the most threatened groups of plants out there. So, like I said, this specialized nature of orchids is one of the threats they face. Pollinator relationship breakdown can happen easily if you've specialized on a certain pollinator and its numbers start to decline. Um, they can also face challenges in reproduction and recruitment, meaning that sometimes it's challenging for them to set seed. But even if they do get pollinated and develop seeds, there's no guarantee that these seeds are going to be able to successfully mature into the next generation of plants. Changes in the mycorrhizal communities that these orchid seeds require to germinate could be one reason for this, but they also require pretty fine fine-tooth like habitat requirements. So they really need these specific microclimates to thrive. They're not as tolerant as some other types of plants are to drought or changes in temperature. And so there have also been studies done that show that orchids are going to lose more of their range to future climate change than other groups of plants might. Out here in the West, they're also suffering from changing fire regimes. This doesn't necessarily threaten Spiranthes infernalis at this moment in time, but a lot of montane region, Western US orchids aren't really fire adapted like some of the other species in those areas are, and so they really struggle in the wake of burns. All orchids struggle with habitat loss and disturbance. Like I said, they require a pretty complicated network of symbiotic relationships to successfully reproduce and survive. And so even little changes can really make a habitat no longer suitable for them. And unfortunately, due to their nature as these charismatic plants that attract a lot of attention, they're vulnerable to poaching and trampling. People want to go visit them, people want to grow them. And the showier and more charismatic an orchid is, the more likely it is to suffer from poaching as a species. This is something that we can do things about as botanists and naturalists and enthusiasts of orchids, which is being careful about, sharing the location of orchids we know, posting them on iNaturalist and that type of thing. You want to make sure you're not putting location data on these poaching prone plants out on the internet. And this goes for other charismatic plants as well. Like if you're a fan of the California native Dudleyas, you don't want to be sharing their location either. These are plants that are likely to be poached. This can be a problem for some cacti species as well. Really just something that unfortunately plagues a lot of our more charismatic plant groups. We can do a lot to protect orchids in addition to protecting them from poaching and making sure we're mindful about how we visit them and who we share their location with. There are both in situ and ex situ conservation efforts ongoing at a lot of institutions to protect our native orchids. In situ conservation efforts kind of address learning about and protecting the plants in their natural habitat. So this involves things like censusing and monitoring plants in their natural habitat, protecting their habitat, both through legal advocacy and action and through land management and restoration. Ex situ conservation efforts have to do with conserving the genetic diversity of a species outside of its natural habitat. So this can include things like seed collection and seed banking, propagation efforts, and establishing living collections of organisms outside of their habitat where the diversity can be protected. So I'm hoping that my research with Spiranthes infernalis will incorporate or contribute to both in situ and ex situ conservation of this species. So my objectives are as follows. I'm hoping to use molecular data, so genetic sequencing on the species to better define the population structure of the species where it occurs, and also to help identify those Spiranthes orchids I was telling you about in Eastern California. I'm hoping to collect and seed bank material from all three Spiranthes occurrences and help develop protocols for ex situ in vitro propagation with some of my collaborators at the Desert Botanic Garden. I'm hoping to survey for additional Spiranthes infernalis occurrences, collect vouchers, and conduct demographic and life history studies in situ on these plants. And once I've done all of these steps, I'm hoping to publish my findings and also help with writing and implementing a conservation plan and pursue legal protections for the species. So, what are the conservation uses of this kind of research? Why would we want to do something like this? One is that the data that we gather on this species can create a better informed petition for listing Spiranthes infernalis under the Endangered Species Act, which could give better protection not only to the species, but also the delicate ecosystems that it exists in, both at Ash Meadows and Railroad Valley. Understanding better the relatedness and biodiversity of these Spiranthes populations will also help better inform the in situ and ex situ conservation actions that we take. Knowing where the most genetic diversity is for a species, understanding how closely the different populations are related for each other, this type of information kind of changes how we'll interact with the different populations. And then also identifying all of the populations accurately is really important too. So those plants in Eastern California at Fish Slough, if they were determined to be Spiranthes infernalis, that would represent a range extension for the species. It would also be the first documented occurrence of the plants in California, and it could lead to better protections for the Fish Slough area because the species would qualify for a California Native Plant Society rare plant ranking as a result of this discovery, and then would be able to be considered under the California Environmental Quality Act. So it's really important that we have these plants accurately identified to really be able to give them the protection that they warrant. I've been undertaking some field work in support of these research objectives, so that's included censusing the known populations of Spiranthes infernalis, surveying for new locations of plants on Bureau of Land Management land around the Ash Meadows National Wildlife Refuge. I've also surveyed for new occurrences in Railroad Valley. I've collected tissue samples at all of these locations for my genetic research, and then I've also been helping with seed banking the species as well. So working with Spiranthes out in these alkali flat habitats can be a challenge. There are these really expansive wetlands that can be challenging to traverse and even more challenging to search for small plants in. As you can see, there's a lot of really dense vegetation in these areas, and Spiranthes can be pretty hard to identify. The picture in the center here is actually what a Spiranthes orchid looks like before it's quite opened up its flowers. I affectionately refer to this as the tadpole phase when they kind of just are a little green blob with no real strong identifying orchid traits yet. And this is what they look like for a good couple weeks before the narrow window that their flowers open. So it can be really challenging to hunt down Spiranthes in situ, especially if it's not a known population. One tool that we've been using recently to help guide our fieldwork is Sentinel-2 satellite imagery, which shows soil moisture from above and is just a really good way to identify at a distance what areas will have the appropriate amount of moisture to actually support Spiranthes in situ. And we can kind of try and match like the imagery that we've seen on existing populations and what the soil moisture level looks like from above at spots where we know Spiranthes grows, and then kind of look for similar moisture signatures in other nearby areas, and then target those spots for our surveys. And so Sentinel-2 satellite imagery has been really useful for Spiranthes surveys. And for those of you familiar with some of the other rare plants in the Amargosa, it's also been used for other taxa like chloropyron and Zeltnera as well. Using this type of satellite imagery and just going out and doing these surveys has resulted in the discovery of a couple new occurrences of this species. We found two new occurrences just outside the boundary of Ash Meadows National Wildlife Refuge, Purgatory Springs and Carson Slough. Sentinel-2 data has also helped us locate two new occurrences in Railroad Valley as well. Another type of fieldwork that I've been engaging in to support the conservation of this species is seed collection, which is just about as challenging as finding these plants in situ is. So orchids, as I mentioned, have these really tiny seeds, and there's also an incredibly narrow window between when the seeds are mature and when they're dispersed. So once seeds reach maturity in orchids, probably within a week or so, the capsule around them will kind of dry out and dehiss, meaning it kind of just opens up and allows the seeds to just be carried by the wind across the habitat, which is really great for the dispersal of the species in its environment, but is really challenging if you're trying to collect some mature seed before they're all gone with the wind in the most literal sense of the word. Um, that's also a good problem to be having in the first place that these seeds get to the point of dispersal. Unfortunately, due to the extreme nature of the climate that these plants grow in and the fact that these groundwater regimes are in some cases already changing, plants in drier areas I found oftentimes will desiccate before the seeds even get the chance to mature. So you'll go out and find a really nice population of plants and come back a couple weeks later expecting to be able to collect seed. And unfortunately, the plants will kind of just have shriveled up and dried up instead of producing seed. There's also sometimes other issues afoot, like insect herbivory on seed capsules, where you'd be hoping to collect seed, but someone else beat you to it, and it's a bug. Um, and just guidelines and protocols to keep in mind too when you're out here collecting these seeds. So the Center for Plant Conservation recommends that to avoid destabilizing a population's natural ability to disperse seed and recruit the next generation of plants, you don't collect any more than 10% of the seed from a given population. And then also with Spiranthes having that selfing or apomictic breeding system potentially, you really have to make sure you're collecting from a lot of different individual parent plants to get a good amount of genetic diversity, since there's a real chance that all of those seeds in the same capsule are going to be incredibly genetically similar or even identical. So, one of our goals with seed banking and seed collection is to capture as much of the genetic diversity in a population as we can. And so, you really have to make sure you get a lot of what we call maternal lines or collecting seeds from a lot of different plants to capture a good amount of diversity. So, when I go out to collect Spiranthes seeds, I'm shooting for as many maternal lines as I can get, up to 50, which isn't always possible depending on how successful of a year the population has had. And then I collect them all individually and store them as these maternal lines, which for species like this, where the genetic diversity is captured more so between individuals than within individuals, is going to be favored over something like bulk collecting, where you just take seeds from a bunch of individuals and store them together. Despite these challenges, we've still been able to get a good amount of maternal lines from Spiranthes infernalis seed banked this past fieldwork season. We were able to collect 9 maternal lines from the Crystal Reservoir subpopulation in Ash Meadows and 47 maternal lines from the South Spring Meadows population in Ash Meadows, which is the type locality of this species. We got 50 maternal lines from Railroad Valley that were collected around the Chimney Hot Springs area. And then we got 6 maternal lines from Carson Slough on Bureau of Land Management land just outside of Ash Meadows. Another type of work that I've been doing in support of these research objectives is sequencing DNA work. And this actually is done in-house in the CalBG lab. So I'll collect tissue and dry it in silica out in the field, bring it back to our lab, and just do a basic CTAB extraction protocol to get the DNA out of the tissue. After that, we'll do a library preparation for a protocol called ddRADseq that I'll explain in a second. We send it out to get sequenced by our collaborators at UC Riverside. And then once we get those genetic sequences back, we can analyze that data using R packages for population genetics and phylogenetic analysis. ddRADseq is like a newer, about a decade and a half old at this point, methodology for analyzing genetic sequences, and it is full name is double digest restriction site associated DNA sequencing. So for this type of protocol, the DNA is sliced up by enzymes, and the pieces of DNA that are created are then attached to ID barcodes and size selected before sequenced across many different SNPs, single nucleotide polymorphisms, which you can basically think of as locations in the genome. And then we can analyze that data, like all those different little polymorphisms, the different genetic base pair that's present at each of these sites. So this method is really good for non-model organisms like Spiranthes infernalis, where we don't have a reference genome available, and we just kind of want to take a more random sequencing throughout the genome of the species. So what is the value of doing this type of genetic research when we're trying to conserve a species? Why would we do this? Um, Doing this statistical analysis can give us an insight into the genetic diversity and relatedness of the different occurrences of the species. And this is a method that's often utilized in rare species or species with unusual distributions. And Spiranthes infernalis is both. It's only known from two or maybe three sites. They're incredibly separated, and there aren't that many plants left in the world. And so it really does raise the question of how much diversity is left in this species. How recently has there been gene flow between these really disjunct populations? How related are they still at this point? And doing these population genetics analyses allows us to quantify the genetic diversity left in the species, within the populations, and between the populations. It also can give us some insights about whether or not inbreeding is an issue in the species and what the population structure looks like. So, there are a couple different types of analyses that I've run so far on the sequences that we've been able to produce from the genetic extractions I've done on Spiranthes and infernalis tissue. And the first one is a PCA. So, what is a PCA? PCA stands for Principal Component Analysis, and it's where you use a computer program to transform data onto a coordinate system where the axes of the coordinates represent the principal components in the data that contribute most significantly to the variation between the data. This allows us to visualize the genetic structure of the occurrences and the different species. In this case, we're kind of working with one species, but it can also be used to help differentiate between closely related species or maybe species where their validity taxonomically is being called into question. And so when I did my analysis, I was mostly just trying to figure out how related or how similar or different the different occurrences of Spiranthes infernalis was. And as you can see here in a preliminary analysis that I ran, all these Ash Meadows subpopulations that I had discussed earlier and shown you all on the map kind of intermingle here on the PCA. So there's not really great differentiation happening between the subpopulations, suggesting that gene flow is still occurring between them, likely via that wind dispersed seed mechanism that I was talking about earlier. But there is a pretty significant differentiation between the Ash Meadows plants and the Railroad Valley plants, which is exactly what you would expect given the fact that these plants are now separated by 150 miles of unsuitable habitat. Another type of population genetics analysis that you can do with dd-rad-seq data is a pairwise FST comparison. This is a fixation index that you're calculating that shows population differentiation. So a high FST value shows that there's strong genetic differentiation between two populations that you're comparing, whereas a low FST shows that the two populations you're comparing are genetically similar and less differentiated. So what this big complicated mess here is it's basically a grid where you're showing the comparisons between every potential pairing of populations that we researched at this point in the project. And so the main takeaway here is that the Ash Meadows populations were very similar to each other. Um, the Railroad Valley populations were very similar to each other. And if you compare a population from Ash Meadows to Railroad Valley, you get a pretty high FST showing strong genetic differentiation. So this essentially confirms what we had already been able to visualize in one way on our PCA plot, which is that there's low genetic differentiation between the subpopulations at both of these sites, but pretty significant differentiation between the different sites. A structure plot shows basically the same thing. This is another type of statistical metric that is used to show population structure with this dd-rad-seq data. The K value that is displayed here kind of represents the number of clusters that you're looking at, so how many parts you're dividing your plants into. And the program that I use tests to see which K value is the best fit for the data. So in this structure plot, k equals 2 was pretty clearly the best value. And this is expected given the massive disjunct between the Railroad Valley and Ash Meadows plants. So the best way to divide up these plants is kind of into a group of two, which really tightly aligns with the Ash Meadows and Railroad Valley split, showing that there's not a lot of overlap between the two at this point. I also ran some preliminary population genetic statistics on my sequence data. So these are, again, it's kind of like an indecipherable group of numbers, and I do apologize for that. But essentially, what we're calculating here is heterozygosity, so the amount of genetic diversity at these populations, and then also FIS, which is an inbreeding coefficient, which again will show how much inbreeding compared to what would be expected in a neutral situation is occurring in these populations. So overall, heterozygosity is pretty low in these populations. It's slightly under what would be expected in Railroad Valley. And at the type locality in Ash Meadows, it actually does slightly exceed expectations. So the type locality is a little bit more diverse than some of the other places where you can find the species around the wildlife refuge. Inbreeding coefficients, on the other hand, show that there's actually a little bit less than expected inbreeding in that type locality, again, where heterozygosity is doing well. But there are a couple places where inbreeding is a bit more of a problem, such as in the smaller populations to the north of the Ash Meadows Refuge and in the smaller subpopulation at Railroad Valley near Terrace Hot Springs. What about those fish slough plants that I had mentioned earlier? We don't really know what they are, and so we can try and use genetic analysis to get at the question of who are these plants. To do that, I did some sampling of other closely related Spiranthes species in the American West to try and kind of place those fish slough plants in the phylogeny. So, with tissue samples from Spiranthes infernalis, its closest relative Spiranthes porifolia, and then also Spiranthes romanzoffiana and Spiranthes stellata, which are two of the more closely related species in the Western US. I kind of tried to start putting back together a reconstruction of the phylogeny of the Western US Spiranthes. So there has already been some research done on the relationships of these Spiranthes species out west. The most recent publication being a 2019 paper by Matthew Pace and Ken Cameron that used chloroplasts and nuclear ITS sequences to kind of determine how these plants were related to each other. The pink stars on these two phylogenetic trees show where Spiranthes infernalis is found, and you can see that it's most closely related to Spiranthes porifolia, which is the other potential ID for these fish slough plants. So I kind of wanted to sample more heavily from porifolia and infernalis than was done at the time this paper was produced, and to include those fish slough plants to hopefully find out where the fish slough plants, whether it's more closely related to porifolia or more closely related to infernalis, and then have some outgroup sampling to kind of serve as a reference point when I'm looking at the relatedness of these three groups. So I collected a bunch of tissue samples from other Spiranthes species around the western US, especially focusing on Spiranthes porifolia. And then I've started working on a preliminary phylogeny to try and determine what the fish slough plants are most closely related to. So the good news is that the Ash Meadows and Railroad Valley plants are pretty clearly forming these related groups right now, which is good for their status as a species, but does raise the question of whether at some point we could define the Ash Meadows and Railroad Valley plants as separate species, since they kind of form their own clades pretty tidily. And then the fish slough plants are right now kind of falling out in between Spiranthes porifolia and Spiranthes infernalis. So I'm going to keep working on this phylogenetic tree. It's definitely very far from a finished project at this point. And so I need more outgroup sampling from Eastern US species and maybe some more coverage within Spiranthes porifolia to kind of figure out where these fish slough plants really do belong. With that said, this project continues through 2026. I've already done a significant amount of fieldwork this year. I've gone and sampled seed collections from Railroad Valley and Ash Meadows. I was able to collect some ex situ Spiranthes infernalis plant tissue from the Desert Botanic Garden, which hopefully will get a sense of maybe, what kind of breeding system is at play with these plants. If they really are apomictic, you would expect that these sibling plants that the Desert Botanic Garden had propagated will essentially be clones of each other. Otherwise, they will just look like siblings if it's more of a classic selfing methodology. I'm still working on generating new population genetic statistics and improving my phylogenetic tree to get a better sense of where those fish slough plants really fall in the scheme of things. And then I'm in the process of writing, so I'm hoping to publish these results soon, and then also help with the development of management plans and listing petitions for the species. I'm working with a lot of really great organizations on this project. Obviously, we're all here for the Amargosa Conservancy today, but they're protecting a lot more biodiversity in this region than just my study species. They work with other plants in the area, they work with important mammals and amphibians and insects. And so, if you're interested in supporting conservation in the Amargosa, fighting back against some of the threats to the desert that I mentioned earlier in this presentation, I would definitely recommend checking them out. If you're interested in learning more about native orchids in the United States, I can't enough recommend NAOC, the North American Orchid Conservation Center. They have a great website called Go Orchids that's really accessible for anyone who's interested in learning more about orchids, even if you don't have much of a botanical background. And they have a lot of really lovely pictures and fact sheets on our native orchids there. And then finally, I of course have to shout out my home organization, the California Botanic Garden. The grad program I'm a part of, the lab where I do my research currently, and where I'm getting all my mentorship and the wealth of knowledge that's enabling me to do this research. And we don't just work in the Amargosa region, we're really trying to protect native plant diversity all across California and the Mojave Desert and the Great Basin Desert. So check out California Botanic Garden if you're more interested in plant conservation in the American West as a whole. And with that, I'd like to thank you guys so much for joining me for this talk today. I have a list of all of the organizations that have helped fund my research up here on the board, and I'd also like to thank my advisor, Dr. Naomi Fraga, for all of her help with this project. And with that, I think I have a little bit of time to open up the floor to questions.
Thank you so much, Allison. Yeah, if you want to ask a question, we have a chat box, a Q&A chat box if you want to type it in, or if you want to ask it, I think if you raise your hand, I can unmute your microphone. Um, so we have a few questions already, so I'll go through the ones that are in the chat. Okay. Somebody asked, could you please tell us whether it's a monopodial or sympodial?
Oh, that's a good question. I would probably describe our native orchids more as sympodial in nature because it happens more underground. So monopodial and sympodial is a term that's applied to tropical orchids that are grown a lot. And with them, because they're epiphytes and they grow on top of trees or in these kind of open bark media when you're growing them in your home, you can kind of see all the action, right? So the sympodial orchids have a rhizome that goes along and puts out a new growth each growing season. And I would say our native terrestrial orchids tend to kind of operate like that, but it's happening underground. And so they're making each season, Spiranthes infernalis makes a new tuber, and then it kind of fills up during the growing season. And then next spring, a new shoot comes off of that tuber. So it's a lot more dense than like a typical homegrown, like tropical orchid would be when it comes to the way that it's growing. But it's pretty sympodial. Each growing season, it's like filling up a new tuber and then making a new shoot off of the tuber. And just like if you make your sympodial orchids at home really happy, they can get new growth leads. I sometimes do get to find Spiranthes that are so happy they're kind of growing in these clumps with multiple inflorescences and new shoots. So yeah, I would say sympodial, but it's a little bit harder to see happening in real time than it would be with an epiphyte.
Okay, wonderful. There's another question. Did you detect clones or genetically identical individuals within populations?
That is not something that I picked up on with my first round of population genetics, but it's also not something that I was running analytics specifically for. So I think for me, part of rounding out my genetic work and research on this species and like the pop gen that I've done would be seeing if I can find like programs or R packages that specifically are kind of out to look for that and see if I can detect any of that. But I wouldn't say it's something I've seen like a really glaring sign of so far, but it's something that I want to do more analytics specifically looking for. There's certainly some concern with, that potentially apomictic breeding system that much like when you're working with a plant that like kind of grows rhizomatously, you have to be careful that you're not actually counting like different parts of that same individual as a new unique plant. With Spiranthes infernalis, we might see this large census population size, but the effective population size could actually be a lot smaller, if some of those plants end up being genetically identical. So it's something I've been thinking about a lot, but I wouldn't say that I have definitive proof at this point that I'm dealing with a lot of clones in these populations.
Okay. Andrew says, I appreciate your slide on pollinators or a lack thereof. Super
Mhm.
interesting. Was any surveying done during the night? I'm curious of the potential for nocturnal pollinators. Thank you for sharing your knowledge with us.
Yeah, I have not spent as much time as I would like to out at night. There's obviously that chance, right? That like this is I haven't noticed a ton of fragrance on the orchids, but it's like a nice like shiny white flower. You look at it and you think, man, maybe moths are into that. And so There's a chance that moths pollinate this, but even if it is receiving pollinator interactions, I still think that the selfing or apomixis is probably the primary mechanism by which seed set is happening. Just because generally in orchid species that I've worked with in the past that aren't engaging in selfing or apomixis, you can tell at the end of the season because you'll see seed capsules developing only on certain flowers that have been hit by pollinators. Whereas with these, the plants that I find that are still in good condition and haven't, died from drought at the end of the season have just 100% capsule development all the way up and down the stalk. And so I really think that even if they are getting effectively visited by pollinators at a time that I've missed, um, I would reckon that there's something else happening there as well to help the seed set occur. And I will say that I've been there early in the morning, like pre-sunrise, and I haven't managed to catch anything happening then. So it would really have to be like a truly nocturnal thing that's happening like late at night for me to have missed it. But yeah, that's certainly a possibility still at this point. If anyone's interested in setting up like camera traps for pollinators, that's something I'd definitely be happy to collaborate on. But that would be kind of outside of the scope of the project at this point, I would say, since I'm hoping to be on the presses by Spiranthes blooming time next year.
Okay. And then another person asks, based on the preliminary results, is Spiranthes infernalis embedded within Spiranthes porifolia?
I would say no. It is kind of portfolio is kind of showing up on a little bit of a grade right now, which I think could be a weakness of where I'm currently at with my analysis and not having like a distant enough outgroup to anchor. One of my hopes for this round of like genetic work and analysis this fall is to get like a Spiranthes cernua or something like that from the East Coast incorporated as my outgroup because right now my best outgroup is Spiranthes romans-offiana, which I think is really just too close to porifolia and infernalis to really be a good outgroup point. Um, but yeah, they're pretty closely related. The Matthew Pace paper left those two species in an unresolved polytomy, and so I'm hoping that I'll be able to get them looking reciprocally monophyletic. But I will say at this point, I'm pretty sold on Spiranthes infernalis as its own species because it is very clearly forming its own monophyletic clade. Like even if it does end up being embedded in porifolia, I would say that it's just like a really strong case of budding speciation, like out in this super reproductively isolated habitat. Um, but yeah, it's not currently embedded in porifolia like in a major way, but porifolia is kind of on a grade um, next to the monophyletic Spiranthes infernalis group. But it's very tightly in its own monophyletic clade. And so really the species that's I'm having more trouble getting to look the way it ought to right now is porifolia.
Got it. Okay, this next question is a couple of questions in one. So, do we know what sort of fungi these orchids are associated with in the Amargosa Basin? And is culturing these fungi also a part of germinating the seeds that you've collected, or can they be germinated without that symbiont?
Okay, those are all really great questions. So we're not entirely positive what fungi Spiranthes infernalis associates with right now. I'm collaborating with Dr. Melissa McCormick at the Smithsonian Environmental Research Center, who's one of the NAOC collaborators that I mentioned earlier at the end of my talk, to try and identify these mycorrhizae. And so I've sent her some root samples that she's hopefully going to get to sequence and culture, and so we'll get a sense of what fungi can be found in the roots of these orchids. She had gotten a Spiranthes infernalis root sample ages ago from Steve Blackwell at the Desert Botanic Garden when he was out there for research. And I don't remember the exact name of the species, but when they had sequenced it, it came out to be the same fungi that's affiliated with the Canelo Hills ladies' tresses, which is another rare kind of desert-y Spiranthes species in Arizona. And she was really shocked by those results and kind of wanted to be able to like verify them with a second round of tissue samples. So I've sent her root samples from Railroad Valley, and I have en route to her root samples from Railroad Valley again and Ash Meadows again to try and hopefully confirm like so she doesn't just have that one sample she had gotten like what fungi this plant is associating with. So it's not really fully a known thing at this point, but It's something we're looking into, and it's something that should hopefully be known at some point in the near future. Culturing the fungi, once you know its identity, is one option for propagating it. And I think a lot of people do kind of prefer that option because there's a school of thought that it's maybe better for like the long-term development of the plant and its ability to be reintroduced into like its native habitat if you're trying to do like an outplanting exercise to have grown it with its mycorrhizae. However, you very much can propagate these orchids without the fungi. So that's called asymbiotic propagation because it's lacking the symbiote. And what you would then do instead is use like a tissue culture lab setup and nutrient media to give the seed what it needs in the absence of the fungi. And so you take these seeds and you do your best to sterilize them and you get them in like a flow hood and onto nutrient media. And then you kind of seal it up. And it should, in many cases, germinate just as it would with the fungi there, but it's getting its nutrients from the media you put it on instead. So it's a possibility. You can do both symbiotic and asymbiotic germination. And if you don't know the mycorrhizal symbiont for an orchid species, you can always start with asymbiotic germination, but there might be some, shortcomings with that methodology compared to using the fungi.
Okay. Has anyone looked at the Spiranthes species that are in the Ruby Mountains of Elko County, Nevada? Do you know?
I am not familiar with those Spiranthes. I know that in like the northwestern part of this state, there's a lot of Spiranthes romanzoffiana. I'm trying to think about where the Ruby Mountains are. I'm unfortunately not a native Nevadan, as you can probably tell by the way I say the name of the state, I've been told. But I know that there's Spiranthes romanzoffiana in a lot of locations in Nevada, and I would have to look at pictures of the ones in the Ruby Mountains to know whether we're dealing with that or potentially something different. But that's definitely something I would welcome communication about. I always like hearing about Spiranthes, so if you have pictures and you want to send them to me, I'd be happy to look at them.
To know. Okay, and one final question we have. Somebody says there are two types of Opomyxis. I don't know if I'm saying it right.
That makes sense.
Opomyxis in general.
I
Could you
mean,
please expand? There's gametophytic and sporophytic. Which one is more common in orchids?
I actually don't know the answer to that question. I'm not much of an orchid reproductive biology researcher, and I haven't had really the chance to get into what's happening at that level with Spiranthes infernalis. I'm kind of more interested in it from like a impact on like the genetic diversity and the population perspective. So I haven't unfortunately even been able to confirm that there is apomixis in this species yet, let alone which type is happening. So I don't know which is more likely to be going on in this plant right now.
Okay, I think that's our final question, unless anyone wants to ask another or type it real quick. We'll give it a second. Regarding the potential Spiranthes that are in the Ruby Mountains, Allison, did you have a contact
Oh yeah,
information if someone wanted to reach out?
I can be found on the California Botanic Garden website, but in lieu of that, my email is Autry, so AAUTRY at calbg.org.
Wonderful. Okay, I think that was our final question. Last
Great.
call.
Thank you everyone so much for attending and for the really exciting questions. I wasn't sure how we were going to fill 15 minutes of question time, but we did.
Yes, thank you everyone, and thank you, Allison. Your present. Oh, somebody has raised their hand. We have one
Oh,
more.
good.
This will be our final question. Okay, Charlie, I think your mic is unmuted if you want to, or you're allowed to ask your question. If you're talking, we can't hear you. maybe there wasn't a question. Oh, okay, well, if there are
Anyone
any final
who's
questions,
been
reach out
listening
to
in today
Allison.
is welcome to shoot me an email if they have questions later. Thank you guys all so much for being here.
Perfect. Thank you, everyone. Bye-bye.